Let's discuss platelets and the blood clotting process. Now, when it comes to platelets, these are not actually cells, they are fragments of cells. Now, this simply means that they are pieces of cells. Now, platelets are produced in the bone marrow. That is the bone marrow. This is the tissue that is found in our long bones. So, what happens is that within the bone marrow, you're going to have cells known as megakaryocytes. Now, when these cells break apart, they produce these hundreds of small fragments of cells, small pieces of cells that form platelets. Now, platelets are different from other cells in that, number one, they do not have a nucleus and they tend to miss a lot of other organelles. And this is because there's really no need for that. The function of the platelets is simply to carry out blood clotting, which is enough with what they have. Let's talk about the actual blood clotting process. And this is a very, very important process because it reduces loss of blood when blood vessels are injured, such as during a cut. Remember, excessive loss of blood, especially more than 2 liters, can lead to severe anemia, even death, if not contained. So, this process involves a series of complex reactions whereby at the end of it, you're going to have a soluble protein that is found in blood, which is called fibrinogen, being converted to an insoluble protein. And the name of that is fibrin. So, essentially, whatever steps I'm going to discuss is simply to ensure that fibrinogen is converted to fibrin. Now, our starting point is going to be this. It's going to be initiated by a damaged blood vessel, such as through a cut. So, when an injury is exposed to air, that starts out the whole process. So, platelets that are normally found within the blood are going to move towards the site of injury. They're then going to release chemicals that will attract more platelets towards that site. The platelets will then burst open, releasing an enzyme that is found within them. This enzyme is known as thrombokinase or thromboplastin. Now, enzyme thrombokinase has two roles. It has two functions. Number one is that it stops the activity of heparin. It neutralizes heparin. Now, heparin is an anti-clotting factor that is present within blood. Now, remember, in normal situations, that is when there's no injury, we do not want blood to clot, to solidify. We want it to flow smoothly, circulate smoothly within the blood vessels. And that is why heparin is there, to ensure that blood does not clot. However, when an injury occurs, we need blood to clot. That means we need heparin to stop functioning at that particular site of injury so that clotting of blood can take place and blood loss can stop. So one of the functions of thrombokinase is neutralizing heparin. Now the other is that it activates the conversion of prothrombin to thrombin. Okay, let me explain that. Now prothrombin is the inactive form of an enzyme called thrombin. Now the reason why this enzyme is in an inactive form is because it shouldn't work unless when needed during blood clotting. So it's going to be in an inactive form called prothrombin. Now, when it's activated by thrombokinase, it's then converted to an active form called thrombin. So essentially what is going to happen is that you're going to have one enzyme activating the conversion of another enzyme. Now, one thing I would like you to note is this. Prothrombin is formed in the liver and it's always formed in the presence of vitamin K. So vitamin K is an essential vitamin in the process of blood clotting. Now moving on to enzyme thrombin. So it has been activated. Now what is its role? So enzyme thrombin activates the conversion of fibrinogen to fibrin. Now remember what I said about fibrinogen? This is a soluble plasma protein that is normally found in blood. Now in the presence of enzyme thrombin, it's converted into an insoluble form called fibrin. So fibrin then forms fibers that entangle together, that join together to form this meshwork around the damaged part. Now this meshwork is very important because it traps red blood cells at the damaged point to form a clot. When the clot dries up, it forms a scab that stops the bleeding and prevents entry of microorganisms. Remember, if you have an open part of the skin, it's going to serve as an entry point of microorganisms into the body. We do not want that. So the clot dries up, it forms a scab that stops the bleeding and prevents entry of microorganism. Now you might be wondering, why are we converting some of the substances from one form into another? Why not have them ready in their activated forms. Now, I'm going to explain this by stating that blood clotting occurs on a need to basis. That is, when an injury has taken place and there's blood loss, then that is when clotting of blood needs to take place. We do not want blood clotting in normal cases. And that is the reason why fibrinogen is only converted to fibrin when blood vessels are injured. Otherwise, if it were in the form of fibrin within our blood, what will happen is that it will form a clot and will end up blocking the blood vessels and interfering with the circulation of blood. One last thing I would like to mention is the role of calcium ions in this process. During conversion of prothrombin to thrombin, calcium ions are required. So let me clarify that. We need calcium ions and vitamin K. Vitamin K is required in the liver for the formation of enzyme prothrombin. Calcium ions are required during the conversion of prothrombin to active thrombin. I hope that has cleared out any misunderstanding for you. By the way, I would like to say this. If you find this to be a complex process involving several steps, I would highly advise you to create a mind map. This is a mind map whereby you actually put the information in a summarized way with in form of a flowchart. It makes it easier for you to remember. Our brains are designed in such a way that it's easier for them to remember images and pictures rather than words. If you would like to have more information regarding how to create mind maps, especially in biology, check out this video of mine. See you next time.